Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Okay, let's unpack this. For decades, really, when we thought about medicines, therapeutic medicines, our frame of reference was the small molecule, simple chemical compounds.
Right. Things you could synthesize, purify, bottle. Your standard pain reliever aspirin is the classic example.
Exactly. But the whole pharmaceutical landscape, it feels like it's undergoing this profound shift, almost revolutionary, driven by biotech.
That transformation is absolutely fundamental. We're seeing this rapid rise, really the dominance now of biopharmaceuticals, or biologics as they're often called.
And it's not just like a new category of drugs, it feels like a change in the whole scale of medicine.
It really is. We're shifting focus from those simple, stable, chemically made small molecules to these incredibly complex, massive macromolecules, huge things.
And massive almost doesn't do it justice, does it, when you compare them? Like, visualize aspirin, tiny molecule, what, about 180 Daltons?
Yeah, minuscule on a molecular scale.
Now, compare that to a modern biologic. Let's take infliximab, used for autoimmune diseases, rheumatoid arthritis, Crohn's.
A monoclonal antibody, yep. And infliximab clocks in around 149,000 Daltons.
Wow. So quick math, that's like over 800 times heavier than aspirin.
Exactly. And this isn't just some, fun fact for a chemistry class. This enormous difference in physical size and also complexity, it introduces completely unique pharmacological challenges.
Problems that old school small molecule chemistry just never really had to deal with.
Right. Problems of structural integrity, stability, how you even deliver them.
So that's our mission for this deep dive then. To get into those core challenges, understand why these massive drugs are so, inherently fragile, why they're so tricky to distribute through the body.
And then, maybe the most fascinating part, explore the really advanced engineering solutions. I mean, it's like a microscopic manufacturing war going on to turn these delicate proteins or even fragile bits of RNA into effective medicine.
All right. Let's start with the basics. The fundamental pharmacokinetic challenge: size and fragility. So the first big hurdle, maybe the defining one with protein drugs, is really grasping that for these complex biologics, structure isn't just important. Structure is function.
Absolutely. You have to think way beyond just the simple linear sequence of atoms. We talk about the 4 hierarchical levels of protein structure.
All right, okay. Break that down for us.
Well, it starts simple enough. The primary structure, that's just the linear chain, the specific sequence of amino acids, coded by the DNA.
Like beads on a string.
Exactly. Then that sequence dictates how it starts to fold locally. That's the secondary structure. Things like the coiled alpha helix or those flat pleated beta sheets.
Okay, so it's starting to get some shape.
Yep. And as that chain keeps folding up on itself, driven by how the different amino acid side chains interact, you get the overall 3D shape of that single polypeptide chain. That's the tertiary structure.
And then for a lot of these big therapeutic proteins like antibodies, there's one more level.
Right. Often, multiple folded chains or subunits have to assemble correctly, like Lego bricks clicking together. That final assembly is the quaternary structure.
And here's the critical point for pharmacology, right?
Right.
The drug's actual therapeutic activity, its ability to recognize its target, bind to it, neutralize something, depends entirely on maintaining that precise final 3D shape.
Entirely. Especially those tertiary and quaternary shapes. It's the exact conformation, the shape of the binding site, that acts like the key fitting into the body's lock.
So what happens then if that structure gets even slightly messed up? Why are these things so fragile compared to, like, sturdy little aspirin?
Well, they're fragile because that complex 3D shape is held together by relatively weak forces, hydrogen bonds, hydrophobic interactions, things like that. So any slight physical stress or chemical stress.
Like changes in temperature or pH?
Exactly. It can cause the structure to drift. We often see these subtle changes called microheterogeneities, like deamidation or oxidation on certain amino acids, which changes the local charge.
Or sometimes it's more dramatic.
Oh, yeah. You can get changes in the quaternary structure where the individual protein units just start clumping together, aggregating, forming these useless clumps.
And once that happens, aggregation or denaturation, losing that 3D shape.
The binding site is gone. The key is bent, twisted out of shape. You get a rapid, often complete loss of therapeutic activity. You've got this massive, expensive molecule that's now just inert, useless.
And sometimes even worse.
Sometimes worse. It can even become immunogenic, meaning the body might mount an immune response against it.
Okay, that fragility really drives home the point. Let's use the classic case study.
Yeah.
Why can't insulin be a pill? Insulin's a protein drug, right? Relatively small, but still a protein.
It's a perfect illustration. Insulin's a peptide hormone, 51 amino acids. And it absolutely needs that precise 3D shape to dock perfectly with the insulin receptor and, kick off the process of glucose uptake. It's a beautiful lock and key mechanism.
But if you try swallowing that key,
the body immediately sees it not as medicine, but as food, protein, ready for digestion.
So walk us through that destructive process. Where does it start?
Right away, in the stomach. The human stomach is incredibly acidic. The pH can drop down to 1.5, sometimes even lower.
That's really acidic.
It is. And that intense acidity is a powerful denaturing agent. Think about what happens when you cook an egg white. The heat denatures the protein. Acidity does the same thing to insulin's delicate tertiary structure. It unfolds the protein chains, exposes the backbone.
Okay, so the acid melts the key, and then the body attacks again.
Absolutely. Once those chains are linearized, unfolded, they're exposed to the body's highly specialized digestive enzymes, the proteases. In the stomach, you've got pepsin.
And then in the intestine.
As the denatured stuff moves into the small intestine, it gets hit by even more powerful enzymes like trypsin and chymotrypsin. These are incredibly potent tools evolved specifically to break down large proteins from our food.
So they just chop it up.
They rapidly cleave those exposed peptide bonds. They basically hammer the unfolded insulin chain into tiny inactive fragments, individual amino acids, essentially.
So, melted by acid, hammered by enzymes, it never gets across the gut wall intact, let alone into the bloodstream to do its job.
Precisely. And this extreme instability, this rapid degradation, is the fundamental reason why biologics, pretty much across the board, need parenteral administration.
Meaning injections or IVs?
Right. Intravenous infusion, intramuscular shots, subcutaneous injections. We have to bypass the entire gastrointestinal tract at its digestive machinery to get that intact, folded drug directly into the circulation.
Okay, fragility handled. Let's pivot to the second major hurdle, distribution. Let's go back to our comparison, aspirin versus infliximab. This really highlights the pharmacokinetic differences, how the drug moves around.
Yeah, it's a great comparison. Think about aspirin first. It's small, like we said, and crucially, it's lipophilic fat soluble.
Which means?
That combination means it faces almost no barriers. It rapidly and widely distributes throughout the whole body. It slips across lipid cell membranes easily to reach targets inside cells. Pharmacologists call this a high volume of distribution. It gets everywhere.
Okay, now infliximab. You called it a biological supertanker. How does that sheer mass change its distribution?
It changes everything. At 149,000 Daltons, it's massive. And being a protein, it's generally hydrophilic water-soluble. It is physically too large to easily move between different tissue compartments.
So it can't just slip across cell membranes like aspirin?
Absolutely not. It cannot passively cross cell membranes. It's basically trapped.
Trapped where?
Largely confined to the vascular space, the blood vessels, and the immediate fluid just outside them, the extracellular fluid. This results in what we call a small volume of distribution. It often approximates just the plasma volume itself.
Like a huge ship stuck in the main harbor can't get into the small canals.
That's a good analogy.
But wait, infliximab needs to get out of the blood vessels to work. If you're treating Crohn's disease, the target, TNF-alpha, and the inflammation are in the gut wall. How does a molecule that big actually get out?
It relies on a slow process, basically leakage or convection. In healthy tissues, the capillaries, the smallest blood vessels, are pretty tight, not very leaky. Okay.
But in areas of chronic inflammation, like in the gut wall in Crohn's disease, Those capillaries are often structurally messed up. They become leaky, they have gaps.
So the giant infliximab molecule can slowly squeeze out through those gaps.
Exactly. It has to slowly leak out through these gaps
Yeah.
in the inflamed vasculature to reach the site where it's needed.
That sounds inherently slow and maybe inefficient.
It is. That slow, limited distribution, dictated purely by its size and inability to cross membranes or actively target tissues, that's still a major pharmacokinetic hurdle.
So you might need really high doses in the blood just to get enough leaking out to the target site.
Precisely. It means high systemic doses might be needed just to achieve a therapeutic concentration locally. That's a challenge. The small molecules, with their high volume of distribution, just don't really face.
Okay, here's where it gets really interesting, I think. We've set up the problems. The drug's too big, it's fragile, the body tries to destroy it or trap it.
Yeah.
So you can't just, synthesize it and stick it in a vial. You have to engineer the delivery itself. Yeah, this is where sophisticated solutions come in. Designed to boost stability, but also, crucially, to extend how long the drug actually sticks around in the body, its circulation time.
And we're moving beyond just basic chemistry here.
Absolutely. This is advanced materials engineering territory now. One of the first major strategies was chemical modification. Basically tricking the kidney into not filtering the drug out so fast.
How do you do that?
The most common technique is called pegylation. You covalently attach the drug molecule to a non-toxic polymer called polyethylene glycol, or PEG.
Okay, so you stick this PEG chain onto it. What does it actually achieve apart from making the molecule even slightly bigger?
Well, it's a clever trick that exploits how the kidneys work. The kidneys filter stuff out of the blood based largely on size. Generally, molecules under about 60,000 Daltons get efficiently filtered by the glomerulus and rapidly excreted in urine.
Ah, so by attaching these PEG chains.
You dramatically increase the hydrodynamic size of the drug complex, basically, how big it looks to the kidney's filtration system. You can often push it well over that 60,000 Dalton threshold.
So by making it look bigger, you help it bypass that kidney filter, which means it gets cleared more slowly and sticks around in the body longer.
Exactly. It decreases the clearance rate, significantly extends the drug's duration of action. Perfect example is filgrastim. It stimulates white blood cell production, normally needs daily injections.
Right.
Pegylate it, and it becomes Neulasta. Much longer effect, you can give it just once per chemo cycle, for instance. That's a huge difference in convenience and efficacy, all from this chemical disguise.
Okay, so chemical tweaks are one way. But we also rely heavily on physical carrier systems, right? Like encapsulating the fragile drug, the key, inside some kind of protective shell.
Yeah.
Tell us about liposomes and nanoparticles.
Yeah, liposomes are fascinating. They're basically microscopic bubbles made of lipid bilayers, like tiny artificial cells almost. They can encapsulate a water-soluble drug inside their watery core.
And nanoparticles.
Polymeric nanoparticles use different kinds of biodegradable polymer structures to achieve a similar protective encapsulation. But just protecting the drug isn't enough. These carriers also have to survive the body's immune surveillance.
Ah, which brings us to the stealth approach, I imagine.
Exactly. A really key invention was the stealth liposome. This involves coating the outside of the liposome with, guess what? PEG again, that same polymer.
So PEG does double duty. Makes drugs bigger for the kidneys and makes carriers invisible.
Pretty much. This PEG coating acts like an invisibility cloak. It prevents the body's main clearance system, the reticuloendothelial system, or RES, which is mostly macrophages in the liver and spleen, from recognizing and gobbling up these particles right away. It lets them circulate longer.
Okay, so we've made the drug survive the enzymes, survive the immune system, extended its circulation time. Now, how do we get it specifically to the target tissue? This often involves passive targeting, right? Exploiting the disease itself.
Yes, the classic example of passive targeting is the enhanced permeation and retention effect, the EPR effect. This is the phenomenon that allows these circulating nanoparticles to build up preferentially in tumor tissue.
How does that work? Is it like the leaky capillaries again?
It is, but amplified. Tumors grow rapidly, often chaotically. They tend to develop really irregular, hyperpermeable, very leaky blood vessels. These tumor capillaries can have gaps hundreds of nanometers wide.
Much bigger gaps than in normal tissue.
Way bigger. This allows these circulating nanoparticles, which are much larger than traditional small drugs, to slip out easily into the tumor microenvironment. And then, crucially, tumors also tend to have very poor lymphatic drainage. The system that normally clears fluids and particles out of tissues doesn't work well.
So once the nanoparticles leak in, they get trapped.
Exactly. They leak in easily because the vessels are leaky, and they can't get out easily because the drainage is poor. So they accumulate there passively just because of the weird architecture of the diseased tissue.
That accumulation based on tissue problems leads us nicely into classifying the precision of delivery, moving through different levels of targeting.
Right. We usually talk about 3 orders. We start broad with first-order targeting. That's just directing the drug complex generally to the capillary bed of a specific organ or tissue. You're aiming for the neighborhood.
Okay, then.
Then you try to get more specific with second-order targeting. Now you're aiming for a specific cell type within that tissue, like targeting cancer cells that overexpress a certain receptor while trying to spare the normal cells right next to them.
And the ultimate level of precision.
That would be third-order targeting. This involves not just getting to the right cell, but getting inside the cell, and even more specifically, releasing the active drug at a designated intracellular location.
Like inside a specific compartment within the cell.
Exactly. For example, designing the system so the drug only releases its payload after the whole carrier has been taken up by the cell, maybe through receptor-mediated endocytosis, and then trafficked into the acidic environment of the lysosome. That's highly complex, highly compartmentalized delivery.
Speaking of precise delivery, you can't talk about targeted medicine without talking about monoclonal antibodies. MABs, they were kind of the original hope for the magic bullet, weren't they?
They absolutely were, and for good reason. Monoclonal antibodies are defined by their incredible selectivity, their ability to bind with really high affinity to only one specific target, one specific antigenic determinant or epitope.
Which makes them incredibly useful.
Profoundly useful, both for direct therapy, think cortuximab hitting the CD20 marker on B cells, or trastuzumab binding HER2 on breast cancer cells, and also for diagnostic imaging, like Processint.
So the idea was these are perfect guided missiles. Just attach a toxic payload, aim it at the cancer cell, boom, magic bullet, cure.
That was the dream.
But their large size, which we keep coming back to, introduced a critical physical limitation when you try to use them as carriers. An issue of dosing inefficiency.
Yes. And let's use a really specific case study to hammer this point home, to show the limits of that pure magic bullet idea. Trastuzumab emtansine, also known as TDM1 or Kadcyla, it's an antibody-drug conjugate, an ADC, used for HER2-positive breast cancer.
Okay, so this is where the physics of huge molecules meets the reality of drug delivery. Break down the components of TDM1 for us.
Okay, TDM1 is this engineered construct. It links 2 very different things. First, you have the massive carrier. That's the trastuzumab monoclonal antibody itself. Weighs about 148,000 daltons. This is the homing device, the part that finds the cancer cell expressing HER2. It provides the specificity.
Okay, the antibody is the guide. What's the weapon?
Second, you have the tiny but incredibly potent payload. This is DM1, a derivative of maytansine. It's a highly effective cytotoxic agent, a cell killer, and it weighs only about 738 daltons.
Wow, 148,000 daltons for the carrier, only 738 for the payload. That's a huge disparity.
It is. And the success, or rather the efficiency of this whole approach hinges on the ratio between the carrier and the payload. We call this the drug-to-antibody ratio, or DR.
How many payload molecules can you actually attach to each antibody?
Well, because of the complex chemistry involved and just the physical crowding steric hindrance trying to link these things without messing up the antibody's ability to bind, you can only attach a relatively small number of payload molecules to that giant antibody carrier.
So what's the average DR for TDM1 typically?
It averages out to only about 3.5.
Only 3.5 payload molecules per antibody.
At that average, yeah. So every massive 148,000 Dalton antibody is carrying, on average, just 3 or 4 molecules of the actual cytotoxic drug.
That sounds incredibly inefficient just from a mass perspective. Let's actually run the numbers just to really grasp this constraint. If the payload, DM1, is 738 Daltons and the DR is 3.5, what's the total mass of the active drug payload being delivered per antibody?
You multiply them. 3.5 times 738 Daltons. It comes out to approximately 2,583 Daltons.
Okay, 2,583 Daltons of active drug. Now compare that to the carrier mass, 148,000 Daltons.
Right, and when you do that comparison, you reach this really startling, almost unbelievable conclusion about the dosing efficiency.
Go on.
Over 98%. 98% of the total mass that you administer intravenously to the patient is the antibody carrier. It's the delivery truck. It's not the active cell-killing drug.
98%. That's staggering.
Yeah.
You're injecting this enormous amount of biological structure, this massive protein, just to make sure that tiny, tiny fraction of actual payload gets to the target.
It perfectly illustrates the fundamental physical limitation imposed by the sheer size of biologics when used as carriers. It forces us to completely rethink what efficiency even means in targeted medicine. It's not about mass, it's about precision.
Okay, let's shift gears again, now into what feels like the newest frontier. Nucleic acid drugs. These are different again, aren't they? They don't just block a receptor or replace a protein. They go right to the source. They manipulate the fundamental genetic processes inside the cell translation.
Exactly. This whole new class, things like therapeutic DNA, antisense oligonucleotides or ASOs, small interfering RNAs or siRNAs, this really represents the ultimate in targeted medicine in principle. But just like the protein drugs, they face a massive instability challenge, maybe even worse.
Worse.
Because unmodified DNA and RNA are incredibly fragile in the body. They're immediately recognized as foreign or just degradable. They get rapidly chewed up by ubiquitous enzymes called nucleases, which are found all over, in serum, inside cells.
So stability and delivery systems are even more critical here than for proteins. You're trying to smuggle these super sensitive genetic instructions into the cell's most protected areas.
Absolutely paramount. The delivery system is everything. It has to protect that genetic message until it gets precisely where it needs to go inside the cell. Let's look at the first key mechanism here, gene therapy, specifically delivering recombinant DNA or rDNA.
And the ultimate goal of gene therapy, the dream really, is to deliver a working replacement copy of a gene into a patient's own cells.
Right, into their somatic cells, the body cells.
So that those cells can then act like continuous little factories, producing the therapeutic protein that the patient was missing because of a genetic defect.
And the prime example showing the immense power this can have is Zolgensma. It's used to treat spinal muscular atrophy, SMA.
SMA, that devastating inherited disease.
Yeah.
Caused by a faulty SMN1 gene, right? Leading to motor neuron death.
Correct. Zolgensma, which is given as a single one-time IV infusion, delivers a functional copy of that missing SMN1 gene.
Okay, but here's the delivery challenge again. How on earth do you get a relatively large piece of DNA like a gene into the most critical target cells, the motor neurons, which are protected by one of the body's toughest defenses, the blood-brain barrier?
This is where the really clever bioengineering comes in. The functional SMN1 gene, the rDNA, is packaged inside a modified viral vector, specifically an AAV9 vector, adeno-associated virus. It's a virus that's been engineered to be harmless, essentially just a delivery vehicle. And the 9 serotype, AAV9, was chosen very specifically because it has this rare, remarkable ability to actually penetrate and cross the normally highly restrictive blood-brain barrier.
Wow. So the virus itself acts like a biological ferry carrying the gene across the barrier.
Precisely. Once that vector gets across the BBB and targets the motor neurons in the central nervous system, it gets into the cell nucleus. The new functional gene starts getting transcribed and translated, producing the stable SMN protein that was missing.
And that stops the degeneration.
It effectively halts the progressive degeneration of those motor neurons. It's a single treatment that addresses the root genetic cause. Pretty amazing stuff.
Truly amazing. Okay, let's move to the second mechanism. Antisense oligonucleotides, ASOs. These are they're shorter, synthetic single strands of nucleic acid. How do they survive, given how fragile naked nucleic acids are?
They have to be heavily chemically modified. Often, the phosphate backbone is altered through things like phosphorothioate substitution to make them resistant to those nuclease enzymes.
And what's their function? How do they work?
Their function is actually beautifully simple in concept. They are designed to bind through complementary base pairing, like two sides of a zipper meshing perfectly, to a specific target messenger RNA, mRNA sequence inside the cell.
And that binding achieves what?
It can do one of two main things. It can physically block the ribosome from translating the mRNA into protein, basically inhibiting translation, or in more complex cases, it can actually modulate how the mRNA is processed, specifically how it's spliced.
Splicing. Okay, that brings us to the most famous ASO example, probably. Nusinersen, brand name Spinraza, also used for SMA. This is where you see that splicing modulation power, right?
Exactly. SMA patients, remember, they lack a functional SMN1 gene, but they do have a backup gene called SMN2.
But SMN2 doesn't usually work well enough. Yeah. Why not?
The SMN2 gene is almost identical to SMN1, but there's a single crucial nucleotide difference. This difference creates a sequence within the pre-mRNA called an intronic splicing silencer, an ISS.
A silencer.
Yeah.
Sounds ominous.
It is. This ISS region acts like a skip this part signal during mRNA processing. It causes a critical piece of the genetic recipe, known as exon 7, to be routinely skipped or spliced out of the final mRNA blueprint most of the time.
So the resulting protein is incomplete.
Right. The protein made from this improperly spliced SMN2 mRNA is truncated, unstable, and gets rapidly degraded. So SMN2 normally produces very little functional SMN protein.
Okay, so how does nusinersen fix this? It's given intralaphically, right,
Yeah.
to the spinal fluid.
Yes, to get it directly to the motor neurons. The nusinersen ASO is designed to bind with perfect complementarity precisely to that intronic splicing silencer region on the SMN2 pre-mRNA.
So it sticks right onto the skip signal.
Exactly. It acts as a physical roadblock. By binding there, it physically prevents the cell's splicing machinery from recognizing that silencer signal.
It basically masks the skip signal, forcing the machinery to include exon 7.
Correct. It forces the correct inclusion of exon 7 into the final mRNA. The result is the production of stable, full-length, fully functional SMN protein, but now produced from the patient's own SMN2 backup gene.
That's incredible. You're pharmacologically correcting a genetic splicing error with just a short strand of modified nucleic acid.
It's a really spectacular example of targeted genetic modulation.
Okay, finally, the third mechanism, which sounds maybe the most potent, RNA interference, or RNAi, using small interfering RNAs, SIRNAs. This feels like we're actually hijacking the cell's own defense systems.
That's a very good way to put it. That's exactly what we're doing. The siRNA molecule itself is essentially just the guide. It first has to be loaded into a large multi-protein complex that exists naturally in our cells called the RNA-induced silencing complex, RISC.
RSC. Okay, what does the RISC complex do once it's armed with the siRNA guide?
The RISC complex, carrying one strand of the siRNA, basically patrols the cytoplasm like a molecular police officer. It uses the siRNA sequence as a guide to search for any messenger RNA that has a perfectly matching complementary sequence.
And when it finds a match?
Once it finds that specific target mRNA, an enzyme that's part of the RISC complex, a specific protein called Argonaute gets activated. Argonaute is the executioner. It precisely and very potently cleaves or cuts the target mRNA molecule, usually right in the middle.
Cuts it in half. So unlike the ASO, which just blocks something, the RNAi machinery actively destroys the mRNA blueprint.
Exactly. It leads to the degradation of that mRNA. The effect is extremely potent, often called gene silencing, because you're recruiting the cell's own powerful degradation pathway to eliminate the harmful message right at the source before it can even be translated into protein.
And the case study here.
A great example is patisiran, brand name Onpattro. It's used for a condition called hereditary TTR amyloidosis, HATTR. The siRNA drug is delivered specifically to liver cells using a specialized lipid nanoparticle carrier.
And once it gets inside the liver cells.
The siRNA gets loaded into RISC. The RISC complex then finds and destroys the messenger RNA for the TTR protein. By destroying that blueprint, the liver essentially stops producing the faulty, harmful TTR protein that causes the disease pathology. It stops it at the source.
Okay, before we move on from these mechanisms, let's just quickly clarify the difference between ASOs and RNAi again. They both result in genetic silencing, stopping protein production, but they use different machinery, right?
That's a crucial distinction, yeah. Functionally similar outcome, but mechanistically different. The ASO, like nusinersen, achieves its effect whether it's blocking translation or causing degradation, typically by binding to the target mRNA and then recruiting a different endogenous enzyme, usually RNase H.
RNase H.
Yeah, RNase H recognizes the RNA-DNA hybrid structure that forms when the ASO binds to the mRNA, and it's RNase H that then cleaves the RNA strand. The ASO basically flags the mRNA for destruction by RNase H.
Whereas the siRNA.
The siRNA is different. Doesn't recruit an external enzyme. It gets loaded into the pre-existing potent RISC-Argonaute complex. The siRNA acts purely as the guide, and the RISC complex itself contains the enzyme Argonaute that directly acts as the molecular scissors to cut the target mRNA.
Got it. So ASOs call in RNase H. siRNAs use the built-in RISC-Argonaute scissors. Two different enzymatic pathways hijacked for therapy.
Exactly. Both brilliant strategies, but distinct molecular mechanisms.
Okay, so bringing this all together, what is this huge shift in the kinds of drugs we're using, from simple chemicals to these enormous complex biologics, mean for actual clinical practice?
For patient variability, personalized medicine, we've established the big molecules get eliminated differently.
Yeah, that's key. These massive proteins, the nucleic acid drugs, they're typically cleared by mechanisms that don't involve the classic drug metabolizing enzymes. Things like generalized proteolysis, just being broken down as proteins or receptor-mediated clearance, or being cleared by the reticuloendothelial system, the RES, they largely bypass those well-known cytochrome P450 pathways. Right, so given that they bypass the CYPs, why is pharmacogenomics studying genetic variations in those small molecule pathways, the CYP, the phase I enzymes, the phase II enzymes like UGTs, NATs? Why is that still critically important when you're treating a patient with a biologic?
What's hugely important because these advanced therapies rarely exist in a vacuum, particularly in fields like oncology or immunology. Biologics are very often co-administered with traditional small molecule drugs.
Ah, okay. Combination therapy.
Exactly. You might have a patient getting a monoclonal antibody alongside a standard small molecule chemotherapy agent. And that patient's ability to safely and effectively handle that co-administered small molecule depends entirely on their individual genetic makeup regarding those CYP enzymes, UGTs, etc.
And maybe even more critically,
Uh-huh.
let's think back to the antibody-drug conjugates, the ADCs, like TDM1.
Absolutely.
Yeah.
Remember the small, potent cytotoxic payloads that are chemically linked to those massive antibody carriers. Those payloads are still essentially traditional small molecules, often highly toxic ones.
So their fate depends on the old pathways.
Fundamentally, the detoxification, the clearance, and therefore the toxicity risk of those conjugated small molecule payloads depends heavily on phase II enzymes.
Can you give us a specific example? The source material mentioned UGT1A1 and irinotecan's metabolite, SN38. Why is that enzyme so critical?
Right, irinotecan is a common chemo drug. Its active form, the bit that actually kills cancer cells, is SN38. But SN38 is also highly toxic to normal cells, especially in the gut. The main way the body gets rid of SN38 safely is by attaching a glucuronide molecule to it, a process called glucuronidation. And the primary enzyme responsible for that is UGT1A1.
Okay, so UGT1A1 detoxifies SN38. What if a patient has a genetic variation in their UGT1A1 gene?
If they have a common polymorphism, a genetic variant that reduces the activity of the UGT1A1 enzyme, they simply cannot clear SN38 efficiently.
And the consequence of that poor clearance?
The toxic SN38 metabolite builds up in their bloodstream and tissues. This leads directly to severe, potentially life-threatening side effects, particularly debilitating diarrhea and a dangerous drop in white blood cells, neutropenia.
So even if 98% of the ADC you're giving is the big antibody carrier, the patient's genetics, determining how they handle that tiny 2% payload, is absolutely crucial for safety.
Absolutely crucial. It dictates whether they can even tolerate the drug.
And it's not just the metabolizing enzymes, right? We also need to consider genetic variations in drug transporters.
Yes, transporters are proteins embedded in cell membranes. Think of things like OATP1B1 or BCRP. They act like gatekeepers or pumps, controlling how drugs get into and out of cells and into and out of the body.
And variations in these transporters matter too.
Profoundly, genetic variations in transporters can significantly alter the systemic exposure, how much drug gets into the bloodstream and stays there for both co-administered small molecules and potentially even for some of the carrier components or the small molecule payloads themselves. A patient with a faulty OATP1B1 transporter, for instance, might have much higher levels of a co-administered statin, increasing toxicity risk. It affects the whole picture.
So pharmacogenomics isn't just about predicting side effects. It's fundamental to figuring out the right dose and combination for the entire therapeutic regimen, which is often a mix of large and small molecules.
Precisely. And on top of genetics, we absolutely cannot ignore the impact of non-genetic factors.
Like what?
Things intrinsic to the patient, like their actual underlying disease state or the level of systemic inflammation. We know that inflammation, often driven by signaling molecules called cytokines, can actively suppress the activity of drug metabolizing enzymes across the board. A very inflamed patient might process all drugs less efficiently.
And for those fancy engineered delivery systems, the patient's physiology matters directly for efficacy, right?
Absolutely. Think back to the EPR effect for nanoparticle delivery to tumors. That relies entirely on the tumor having those leaky blood vessels. But what if the patient's specific tumor characteristics or the level of inflammation changes that vascular permeability?
Then the passive targeting might not work as well.
It might fail completely. The physical condition of the patient, the state of their disease, directly dictates whether that carefully engineered delivery vehicle can even do its job. It adds another layer of variability.
Okay, this complex web genetics, physiology, massive molecules, engineered carriers, it leads us perfectly to the final hurdle, the regulatory landscape, specifically the challenge of biosimilars. Now, for traditional small molecule generics, the path is pretty clear.
Yeah, for small molecules, it's relatively straightforward. You need to show pharmaceutical equivalence, same active ingredient, same dose, same route, etc. And you need to show bioequivalence, basically, that it results in the same concentration of drug in the bloodstream over time.
And if you prove those two things,
If PE and BE are demonstrated, the regulatory agencies, like the FDA or EMA, allow the assumption of therapeutic equivalence, meaning they assume it will work the same way and be just as safe as the original brand name drug.
but you absolutely cannot make that same assumption for biologics, for biosimilars. Why is getting true identicalness basically impossible for these big protein drugs?
Because the manufacturing process isn't just straightforward chemical synthesis. A biologic drug is produced in or by a living system, a specific clone of cells, often mammalian cells. These cells don't just produce the protein, they fold it, they perform post-translational modifications.
Like adding sugar molecules.
Exactly, like the specific pattern of glycosylation. These complex sugar chains attached to the protein are crucial. They affect stability, how it interacts with receptors, its immunogenicity. And the exact nature of these modifications, these subtle structural features, what we call microheterogeneities, is inextricably linked to the entire manufacturing process. Meaning, even if two different companies start with the exact same DNA sequence coding for the protein, if they use a slightly different host cell line or different conditions in the bioreactor or different purification steps or even different storage conditions, the final product, that complex 3D molecule with all its subtle variations, will have slight differences.
You can't separate the products from the process.
You absolutely cannot. That delicate final structure and its purity depend entirely on the production chain. And because of that, the regulatory standards for biosimilars, which are essentially the generic versions of biologics, had to be completely different from small molecule generics.
You can't just assume therapeutic equivalence based on bioequivalence. So what do they have to show?
A company developing a biosimilar has to demonstrate high similarity to the original reference biologic. This means only minor differences in clinically inactive components are allowed. And crucially, they must prove, through a whole battery of analytical, non-clinical, and often clinical studies, that there are no clinically meaningful differences between their biosimilar and the reference product in terms of safety, purity, and potency.
And that evaluation is much more extensive than just checking blood levels.
Oh, far more extensive. It requires detailed head-to-head comparisons of pharmacokinetics, PK, pharmacodynamics, APD, how the drug affects the body, and very importantly, comparative testing for immunogenicity.
Because those subtle manufacturing differences might make the body react differently.
Precisely. Those slight variations in folding or glycosylation could potentially cause the patient's immune system to see the biosimilar as foreign, leading to the development of anti-drug antibodies that could neutralize the drug's effect or cause other safety issues. The regulatory bar is set very high because the complexity and inherent variability of the molecule demand it.
Wow. What an incredible journey into just layers upon layers of complexity. When you really stop and put that 180 Dalton aspirin next to the 149,000 Dalton infliximab, you realize the whole foundation of pharmacology has fundamentally shifted.
It really has. The central challenge for drug developers has moved almost completely away from just synthesizing a stable chemical compound and towards designing, engineering, and managing this vast, complex, and incredibly fragile biological delivery and manipulation system.
And the solutions we've talked about are genuinely mind-bending examples of ingenuity, aren't they? Whether it's sticking a peg cloak on a liposome to hide it from the immune system,
Or using a modified virus like AAV9 to literally smuggle genetic instructions past the blood-brain barrier.
or hijacking the cell's own RISC machinery with a tiny strand of SRNA to precisely silence a faulty gene.
It demands this radical convergence of fields, advanced chemistry, material science, molecular biology, genetics, pharmaceutical engineering. It's all coming together.
And that final layer of complexity, the one that really makes the biotech world fundamentally different from traditional pharma, is that understanding we reached. The physical structure, the purity, the effectiveness of these big biologics are just so deeply intertwined with how they are actually made.
Which brings us right back around to our final, maybe provocative thought for you to consider. Unlike small molecule drugs, where the chemical formula itself basically defines the product. C9H804 is aspirin, end of story for biologics, the manufacturing process itself.
The specific cell line used, the exact purification methods, the formulation buffer, the storage temperature.
All of that is inextricably linked to the final product's quality, its efficacy, and its safety profile. The process is the product in a very real sense.
So it raises this really profound question, doesn't it, as we move deeper into this era of massive medicine. Is the true drug in the biotech world just the final, enormous, fragile molecule itself? Or is it actually the entire, highly controlled, meticulously validated end-to-end production chain that guarantees that molecule can actually do its job safely and effectively?
That intricate relationship between the process and the product, that's really what defines this revolutionary field.
And definitely something worth mulling over as this technology continues to reshape medicine and human health.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.